Hello, listeners. Yamina here. I'm very excited to have you join us for another Dr. GPCR podcast episode. If you haven't heard, we are officially a nonprofit 501c3 organization. Becoming a nonprofit only solidifies our mission to serve the GPCR community. Therefore, we're taking a short podcast break starting in July to come back with new episodes at the end of August, as well as an amazing set of events in the fall. If you are an ecosystem member, by now you know that every Thursday you get your weekly news or weekly GPCR news delivered directly in your inbox. If you're not an ecosystem member yet, join us today. Thanks to our fantastic team of volunteers of GPCR scientists, a GPCR article will also hit your inbox weekly on Tuesdays. Also, mark your calendars for not one, but two Dr. GPCR University classes with Dr. Terry Kanakin. The first one is titled Techniques for Effective Lead Optimization of Candidate Molecules, Principles of Pharmacology for Drug Discovery, which will start on Thursdays, September 5th at 10 a.m. EST and run for 4 consecutive weeks for 90 minutes. Then, on October 3rd, an advanced class will start about therapeutic translation of GPCR activity, advanced method for the optimization of candidate selection. This class will run for 5 consecutive weeks, also from 10 a.m. Eastern Time on Thursdays. For both courses, we will be offering 5 scholarships per course to GPCR scientists who live and work in developing countries. So stay tuned. Just a short week after the beginning of our second Dr. GPCR University class in the fall with Dr. Kanakin, on October 11th, we will be hosting our Dr. GPCR Symposium on GPCRs as therapeutic targets. During that day, we'll have talks from speakers of all levels, both from industry and academia, a poster session, as well as a panel discussion. We're also excited to be co-organizing our very first in-person Adhesion GPCR workshop in collaboration with Dr. Anthony Boucard from the Adhesion GPCR Consortium. Mark your calendars for October 23 to 25 and join us in Mexico City for a fantastic meeting on all fascinating aspects of adhesion GPCRs and more. If you'd like to sponsor the Adhesion GPCR workshop, please email us at hello@drgpcr.com. Both emails, actually, we have now 2 emails, hello@drgpcr.com and hello@drgpcr.org. Both of these emails will be active for a foreseeable future to make sure that we don't miss your emails. And now, let's dive into this episode. Hello, everyone. This is Yamina Bershish, founder of Dr. GPCR. Welcome to another episode of the Dr. GPCR podcast. I would like to introduce you to my guest. Today, I have the pleasure of talking with Dr. Fiona Marshall. Hi, Fiona.
Hello, everybody. I'm very pleased. Always happy to talk about GPCRs.
Great to have you on the show. So, how are you doing with this whole new era that we live in?
Oh, it's fine. You know, I prefer meeting people in person, but we've got used to doing everything through Zoom and computer type meetings, and a lot of the work that I do can be done through that medium. So we just have to get on with things and carry on as best that we can.
Absolutely. So can you a little bit tell us about your career and what led you into getting interested in GPCRs?
Yeah, so just a bit about my sort of background career. I mean, I grew up in England, although I'm now living in the United States. I studied biochemistry as my first undergraduate degree. That was at the University of Bath. And then I went to Cambridge to do a PhD. I did think about doing medicine, but like anybody who is interested in biological sciences, but I decided I'd rather focus on research into the causes of disease and hopefully look for new treatments. And I first became interested in GPCRs during my undergraduate studies at Bath, and I can still actually remember the exact details of when I first heard about GPCRs, which was in a lecture on how the beta-adrenergic receptor in the heart is activated by adrenaline, how it then engages the G protein and initiates the whole signaling cascade. And I found it absolutely fascinating, and then decided that I would want to work on GPCRs for my PhD. And I wanted to combine that with neuroscience. So I chose to work on neuropeptides that were in the basal ganglia that regulate dopamine release. So neuropeptides like CCK and the neurokinins. And then I went straight from my PhD to join what was then called Glaxo, now GSK. And I started working in the neuroscience team there. And that was on a lot of different projects, but a lot of them were GPCRs. And that included working on neurokinins in pain, for example, melatonin in circadian rhythms. After a few years, I moved from the neuroscience group actually into a group entirely focused on GPCRs. That was called receptor systems. And that was at the time of sequencing of the human genome, so in the late 1990s. And the team made many important discoveries in GPCR research then. That included the identification of the heterodimerization of the GABA B receptor. The team also identified the RAMP proteins that constitute part of the CGRP family of receptors.
Wow.
And we also identified ligands for a lot of orphan receptors. So that includes the free fatty acid receptors, GPR41 and 43, which are now called FFA3 and FFA2. We also identified the receptor for the nicotinic acid, a drug nicotinic acid, and that's now a hydroxycarboxylic acid receptor. So many years then working in that area, I then left GSK and spent some time actually working for myself as an independent consultant and sort of back into academia teaching in Cambridge.
Mm-hmm.
And then, you know, that ultimately led me to co-founding Heptares Therapeutics. Obviously, that's probably what I'm best known for in the GPCR field, now called SOSE Heptares. And, you know, that was a company entirely focused on GPCRs.
And it still is.
Oh, absolutely. Yes.
How fun. I think a lot of people I speak to, including you and I, started working on GPCRs because they attended some lecture where
Exactly.
people were
Yes.
you know, talking about signaling or something like that. I don't know if
is your imagination, and yeah, you want to know more about it.
It does. Absolutely. So you had mentioned that you were at GSK. Were you in a large group? How was life different at a company compared to an academic setting during your PhD?
Well, my PhD at Cambridge actually was interesting because that was what's called a case award. So it was in collaboration with industry, as actually a company called Park Davis, who went on to be part of Pfizer. So they sponsored my PhD at Cambridge. And so I actually split my time. Most of my time was actually spent doing my PhD within the company labs. And my supervisor was John Hughes, who's best known for the identification of the enkephalin peptides. And he was my supervisor, although I also had an academic supervisor. So I was really right from the start very much embedded in that industrial setting and very keen on doing applied research, applying my research to drug discovery. So in a way, it was actually quite a natural transition then to move into the team at Glaxo at GSK.
And then the next transition, which was almost natural, and correct me if I'm wrong, was the idea of, you know, exploiting GPCRs as drug targets and co-founding Heptares. Can you tell us a little bit more about the circumstances in which Heptares was born?
Yeah, so this was really based on the work of Chris Tate and Richard Henderson at the Laboratory of Molecular Biology, known as the LMB in Cambridge. And they were working for many years. In fact, I collaborated with that group while I was at Glaxo, many years looking at different strategies to try and solve crystal structures of membrane proteins. And many of those initially focused on trying to improve expression level. But actually, even when we did that, we got better expression. The protein still didn't crystallize. So Chris Tate really identified that the problem was the stability of the protein when it was in detergent. And he looked for ways in which you could increase stability through mutagenesis. So that came up with the idea of thermostabilization, that you could introduce mutations into the protein, look at the thermostability in detergent, and if you selected mutations that increased stability, you would get a better protein that could be more easily purified and that would then crystallize. So I was actually at that time working, that was part of the time that I was working for myself as a consultant. And a lucky situation is that the venture capital company asked me to go and visit the team at the LMB and see if I thought a company could be formed out of their work. So I went along, spoke to Chris and Richard, and then also Malcolm Weir was another person. He was actually at the MRC at that time in MRCT, and between us, we just thought it was a fantastic opportunity. So I wrote a report back to the venture capital company and said I recommended funding this company, and then I spoke to Malcolm and Chris and Richard and said that I would like to join and help set the company up.
Wow. What a coincidence. You go to meet people and then you end up founding a company.
Yeah, it was great sort of coincidence of the right people at the right time coming together.
Oh, wow. Fantastic. At this point, you're co-founding Heptares. You get funding from a venture capital firm. Were there in the beginning any challenges that you were facing, and can you tell us more about those?
Yeah, so setting up a new company from scratch is always quite challenging. You need labs to operate in. The benefit we had is that we could use the MRC technology incubator labs, so we're able to use their labs, some of their equipment. Yes, you have to raise funding, and that's not just at the start, but as you go on, that's one of the main challenges is finding sources of funding, and then also what is your plan, choosing which receptors to work on. I think we made some very good choices at the start. We focused initially on adenosine receptors and also muscarinic receptors, and we were able to solve structures of those proteins and use that for structure-based design, and those went on to be collaborations with AstraZeneca and with Allergan. So yeah, the initial choice of targets is one of the most important things, and then how you can prosecute those and stay ahead. Of course, you've got to then recruit in all the right people, and we had a lot of people to try and recruit who, not many people actually had any experience of working in membrane protein structural biology. There was a poor success rate before that. So what we actually did was to bring in people who'd worked in other areas, and then we sent them up to the LMB, and they worked in Chris Tate's lab for a while to get a better understanding of how to work on membrane proteins. And we recruited chemistry team, not who'd worked on GPCRs, but actually who'd worked on kinases, and we're used to using structures for structure-based design. So bringing all those people together was one of the things that made it work well.
That's nice. You had in Chris Tate's lab, you had a training to work on GPCRs, which is fantastic.
Exactly.
And it's not trivial, especially when it comes to crystallizing GPCRs. It takes an awful lot of work and an awful lot of time to get these structures.
We continued. Another thing that I think which contributed to the success is that we continued to collaborate with the academic founders, for many years. I mean, all through the company, they're still collaborating together. So that meant that we funded further postdocs and PhD students in Chris's lab that could continue to develop the technology. So the technology didn't stand still. It moved on and kept ahead. And of course, the initial focus was on X-ray crystallography. And then, it moved towards doing more on cryo-electron microscopy as another sort of route to getting GPCR structures. And of course, more recently, that has become very, very successful for getting the complexes with G proteins.
Yes, well, last month, as you might know, I do have a monthly newsletter, and we compiled 10 structures that were published only last month. Crystal structures.
Amazing. It just has kept on going. Yeah, fantastic.
It's been booming, which is great for drug discovery and understanding GPCR function.
Exactly.
From your experience as a founder, co-founder, and CSO of Heptares, were there any lessons that you were able to learn and take with you on further along in your career?
Oh, yes. I mean, I think, the main lesson is getting the right team of people together in anything that you do. And I think that applies to any role. And so one of the things I'm doing now is that I was, it's very sad to leave Heptares, but I was recruited to set up a new drug discovery site for Merck, or MSD, as they're known outside the US. In London, and had the opportunity to start from scratch all over again, to recruit a whole new team, to decide on the areas that we could work on. And so a lot of the experience of how you can put a team together, getting as diverse a team as possible, with different backgrounds, different perspectives, that I think and how you motivate people to do exciting science, then, that's probably the most important thing that I learned at Heptares that I've tried to take forward to other areas.
Wow, that's great. So you mentioned that through very, very early on in your career, you were introduced to GPCRs, and they played an important role throughout your career. Everyone responds yes to this question, but I'm going to ask it to you anyway. Do you think GPCRs are still good drug targets?
Yes, well, of course, I was asked that many times during the raising of money at Heptares, and of course, I always said that, and I still do genuinely believe that GPCRs are excellent drug targets, and this is for a number of reasons. I mean, one thing, that they can be targeted by both small molecules as well as now increasing antibody therapeutics. Obviously, being on the cell surface, they can be considered quite druggable targets. But, having said that, of course, there are some are more difficult to drug than others. All cells express different sets of GPCRs, and some have got, unique expression patterns that allows you to target specific cells. And of course, GPCRs play such an important role in so many physiological and pathophysiological processes. So they're implicated in many different diseases. So, immune regulation, metabolic processes, neurotransmission, and, still only around a third of GPCRs are drugged. So, there's a huge opportunity still left.
Some might say that when it comes to drugs targeting GPCRs, the low-hanging fruit has been picked. What are your thoughts on the most interesting new developments in GPCR drug discovery?
Well, so, I think, the interesting new developments, of course, are still, I'm going to go back to saying the structures, because actually having the structures now is really shedding a lot of new information on how GPCRs operate as a sort of machine, if you like. Yeah. And, I think what was particularly interesting and surprising during my time at Sosei Heptares with the new structures were how many compounds were found to bind to unexpected allosteric pockets on the receptor. So, extrahelical regions, which we found on the glucagon receptor, or C5a antagonists bound there, or drugs that bound on the intracellular side. So those structures are really enabling us to find new ways of drugging GPCRs in different ways, and that could be either existing receptors or new receptors. Another area which I think is quite interesting at the moment is techniques in peptide engineering. I mean, as you know, peptides are often the natural ligands for GPCRs, and peptides can make very good drugs themselves. There's a lot of new developments in how we screen for novel peptide scaffolds. So, for example, cyclized peptides can be screened using phage display. And this is an interesting way of identifying modulators of GPCRs. And also now there are ways of formulating peptides to be orally bioavailable. So again, it gives you sort of new opportunities. And interestingly, even though a receptor, even if it isn't a natural peptide receptor, non-peptide receptors you can still have peptides that bind to them and modulate them. So that's another area which I think is quite interesting.
Yes, but one challenge with peptides could be the bioavailability and the half-life. What are the methods that are used currently to increase this bioavailability? You mentioned that you could have a drug that's a peptide that you could take orally. What are people working on right now?
Yeah, so you can engineer the peptide itself. That's, as I say, making cyclized peptides is one, putting bridges within the peptides, introducing unnatural amino acids or unnatural side chains. So that helps to stabilize peptides, for example, from the natural breakdown mechanisms. But there's also additives that can be given in the formulation that alter the permeation. So they alter the gut epithelial, allow more peptides to actually pass through the gut wall into the bloodstream. So these are the types of strategies that are being used.
All right. Would it be also an option to somehow stabilize a peptide by conjugating it with an antibody?
Yes, that has been done as well. Yes.
Great. So we've talked about the multiple GPCRs that you had worked on. in the current status of GPCR drug discovery, what are the receptors that pharma and biotech are currently focusing on? The sexiest ones.
Yeah, so, I mean, there are many GPCRs, I think, that are still really interesting. I mentioned GPCRs that are in immune cells, and there's a lot of interest really in immunology at the moment, because immunology is playing a role in so many different diseases. We've seen in the oncology area harnessing the immune system, but also we now know that in neurodegenerative diseases, the immune system is playing an important protective role. So, GPCRs that regulate immune function, these are things like chemokine receptors, lipid receptors, adenosine as well. And there's also the MRGX receptors, which are on mast cells. So, I think all of these families are interesting. The other area which of course has got increasing interest is in GPCRs that are activated by metabolites from the gut microbiome. And I mentioned obviously GPR41 and 43 that we originally identified were activated by short chain fatty acids. There's also GPR84 and 120 that are activated by long chain fatty acids, and ones that are activated by citric acid cycle intermediates like GPR91 and 31. So I think there's a huge area of interesting emerging biology coming out in how the host microbiome interactions may be mediated by GPCRs. So they're certainly some of my sort of favorite ones at the moment. But of course there's still all these orphan receptors left, and that's another whole area that's really, really interesting. And I sort of feel that there must be something that we're missing, that there's still, over 100 orphan receptors. We don't know what they're doing or what their role is.
Yeah, and we're still discovering new receptors. Last month, a new GPCR was cloned, which is not something that you'd see every day. It was more in the 90s and 2000s that all these GPCRs were cloned, but I was surprised and happy at the same time to see that there was a new GPCR that was being cloned. And who knows how many other receptors we haven't yet identified.
Exactly.
What I wanted also to ask you is, so you mentioned all of these GPCRs that are interesting, that right now play a role in immunology and oncology, in metabolic diseases. What are the key challenges to drugging these GPCRs?
Well, aside from actually for drugging them, a key challenge for drug discovery, not just for GPCRs, is really to study the role of the target in the context of human disease. And many drugs have failed, including a lot acting through GPCRs, because the validation that we had in animal models didn't translate into efficacy in clinical studies. So, we really need to be much better at studying human biology and relevant human cells. And this is particularly important for GPCRs. For example, we're very excited about biased agonism. It's very appealing, but unless you can really confirm what the proper signaling pathway is and the level of coupling in the relevant human cells in the disease tissue, you could easily make the wrong judgment about your agonist design. So that for me is the biggest challenge, is really understanding human biology.
Agreed. Especially with the biased agonists, with this whole evolution in the field, people for a long time thought it was one pathway versus the other, and when it turns out that it's more complicated than that, especially in a pathological setting, and there is a finesse that needs to be learned about. So agreed, we need to know more about the biology and the pathophysiology and the role of GPCRs in these diseases, but at the same time, combining that knowledge with getting more crystal structures is a nice way to combine this information and further develop drugs targeting GPCRs in a relevant disease context. What are your thoughts about the best way to find allosteric modulators in the context of disease implicated GPCRs?
Yeah, so I mean, to find allosteric modulators, you have to be very careful how you set your screens up. And ideally you want to run a screen both in the presence of the natural agonist, ideally at more than one concentration, because if you put in too high a concentration, you might not see a potentiation with an allosteric modulator. So looking at different signaling pathways, having the screen run in the absence of any other agonist, but then in the presence of the natural agonist ligand, these are going to be the best ways of finding allosteric modulators, but also doing very high throughput libraries like DNA encoded libraries on purified proteins. That allows you to find binders to the receptors that you might not find easily in cell-based assays. And if you can then combine that with getting the structure of those molecules, those allosteric modulators, then you can use structure-based techniques to optimize those.
What would be I don't want to say the best, but what would a good drug look like? Would it be an agonist, an antagonist, an allosteric modulator, or
interestingly, I mean, actually, out of all the GPCR drugs there are at the moment, I did an analysis once, and it's almost exactly 50% agonists and 50% antagonists. So they're both good. Of course, there aren't that many allosteric modulator drugs still, and I think that's because there are challenges in actually translating the level of allosteric modulation into the clinic. So we still need to do more work on that. But, the best drugs are ones that have really strong validation of the role of the GPCR in the disease, and then have all the properties that you need in terms of safety, duration of action, target engagement.
And the minimal amount of side effects that goes with that.
Exactly, of course, yes.
What are the disease areas where new connections to GPCRs are currently being made?
Well, the oncology area is quite interesting because, again, this focus on the immuno-oncology area and the role of immune cells allows you to then identify a number of GPCR targets. And so that's, again, the chemokine receptors are being looked at in that context, the adenosine receptors, which regulate immune cells. So that's one area of interest. Another one is, the proton-sensitive GPCRs. So these are activated by changes in pH. They're receptors like GPR4, 65, 68, 132. These have been implicated in tumor growth and in tissue repair. So they're another really quite interesting family. And then, the family that we still don't really understand much about are the adhesion receptors. We're starting to know more about the fact that they really are GPCRs. They really do signal to G proteins, and a few now examples of some small molecule modulators have been identified. So I think that's one where there should be increasing knowledge coming out. But those do, again, seem to be potentially involved in oncology and in immune function, possibly in development.
Yeah, adhesion receptors are very challenging to study to start with.
Yeah.
So we need to focus more on that. And I recently spoke to a previous guest, and we were talking about taste receptors. And I learned that you have taste receptors in your gut. Potentially, those also may play a role in a metabolic disease or even in cancer.
That's right. Yeah.
More work that needs to be done. In the context of these GPCRs and in these new connections that are made, especially in cancer and oncology, there seems to be a disconnect between, let's say, the GPCR world and the cancer world. And there is a need to better understand the function of GPCRs in the context of cancer. How can we better target these GPCRs in this in the oncology setting?
I think there definitely was a separation of cancer with GPCRs. I mean, I can remember even when I worked at GSK, going to the oncology people and pointing out to them that GPCRs could be interesting targets. And this I'm talking about 20 years ago now. And they were like, no, we're not interested in GPCRs because they're very focused on intracellular signaling at that time within the tumor. But I think now that oncology is thought of more broadly in the tumor environment with all the different cells that are in the tumor milieu, so not just the tumor cells, but also the other immune cells, I think GPCRs are becoming a much more interest into cancer biologists. And of course, they are also on tumors and they are upregulated. So ways where you're looking for targeting the immune cells to the tumor, GPCRs can act as a point of interaction there as well.
Definitely. Do you think the lack of interest in the beginning from cancer biologists towards GPCRs was the fact that in many cases, the receptors are rarely mutated in cancer, but they're overexpressed.
Yeah, they're not oncogenic. Yeah, they can be abnormally expressed. But also GPCRs tend to be more modulatory, I would say. At the time, cancer biologists were looking for targets within tumor cells that would kill the tumor cell. And normally a GPCR drug won't do that. It might increase proliferation or slow down proliferation, but not usually trigger cell death. So that was really one of the reasons why there wasn't that interest. But I think now you can see that enhancing immune cell function allows you to then kill tumors. And so that's the route in through GPCRs.
And also the availability of new antibodies and new ways to quantify these receptors and show that they're overexpressed either in the cancer or in the cancer environment as well also helps make that connection of GPCRs and cancer.
Yes, I think you're right. I mean, this idea about really studying human cells and human tissue applies to all different disease areas. And one technique that we're using more and more now is single cell RNA sequencing and really getting at the expression profiling at the cellular level. You do still have to check that the protein is there or not. It's interesting. Sometimes there can be a disconnect between the RNA presence and protein for GPCRs. So, because sometimes they're expressed for quite a long time at the cell surface, so the levels of RNA can be quite low, even though the receptor protein is actually there.
So you mentioned single-cell RNA sequencing, which is a nice tool to better understand the expression profile of GPCRs, for example, in cancer cells. What are the other tools or initiatives that could speed up the identification of GPCRs in the context of cancer, for example?
Well, I'm going to throw back to the context of neuroscience, which is my area, some of the techniques that we're using. So we're using, again, trying to get human tissue. The nice thing about cancer is you can actually have human tumor cells that are taken out of patients and you can study those. So that's very useful. That's much more difficult in the case of neuroscience and Alzheimer's. You don't usually get bits of brain tissue that you can use. So what's really helped us there is being able to use iPS cells that are being differentiated into different neuronal populations, like dopaminergic neurons, for example. And you can derive those iPS cells from patients who have genetic forms of the disease, so inherited forms of Parkinson's disease, for example. And then you can actually study your GPCRs on those cells and how they signal. So that gives you a different strategy for validating the target biology.
Great. I think having these iPS cells and being able to differentiate them, especially from patients, could be really the best and most direct context in which you can study the function of a GPCR in a disease setting at this moment. What are other key informations that are missing that would help GPCR drug discovery? So today we can, crystallize fairly easily compared to 20 years ago GPCRs. We have all these models. We have great tools to do virtual screens, to do HTS screens very, very quickly and very fast. And we have all these libraries. Is there any key information that's missing to help us move forward?
If I go back to the orphan receptors, we're still totally missing what over 100 receptors actually do, and what their ligands are, how they signal, how they're behaving. And so, there's a huge amount of work still for people to do to unravel these. I think, before we used to have to do knockouts, and almost all of these orphans have been knocked out, but then you don't necessarily see an obvious phenotype in the knockout, so then it makes it harder to link those to disease. What you need to then do is look in more careful detail at different tissues and see what map different changes. Again, you can use things like RNA sequencing and high throughput proteomic techniques to look for changes as a result of changes in the expression of these GPCRs. So that's maybe one thing that will help us to link these orphans to diseases.
Since there is so much more to do and so much work to do to help drug GPCRs in many different disease contexts, what would be your advice to junior scientists who are aspiring to help improve GPCR druggability and understand GPCR biology in general?
Well, I'd say jump right in because, there's plenty to be doing. And I think, people need to think about what excites you most in the area. Are you interested in the disease aspects? Are you interested in new technologies, technology development? It's important, of course, to find good mentors who can help you. And do that by networking as much as you can. For example, going to conferences and joining WebExes. So, at the end of the day, you've just got to do a lot of hard work and do a lot of reading, and that's what helps, you come up with ideas.
And sometimes be a little bit lucky and meet the right people.
Of course, that always helps.
It's a good time right now, although we're living in an totally new world, but it's a good time to go to meetings. There is plenty of virtual meetings, virtual summits, where you can very easily get access to it, and it kind of disrupts that barrier of having to travel to a space, to a new spot to meet people. So I think it's a good time to do that, especially networking online. So what would be your advice to someone or a GPCR scientist who has an idea and would like to turn it into a company, into a product to help improve human health?
Well, again, it's going around and talking to the right people. So try and connect with people who have set up companies before, and initially really try and work on a good plan, business plan of how you would take that product forward. How is it going to be unique from what other people are doing? What do you actually need to get to that? How many people would you need to recruit, for example? Is it something that you can work through contract research companies, so be like a semi-virtual company? So you need to have a good sort of scientific plan, but also a business plan. And then, start to go and talk to people that have set up companies already. At some point, then you would go and speak to usually a early investor that founds startup companies. Quite often for people who are in universities, they have advisors that can help. They can be in the tech transfer offices, for example, or they have entrepreneurs in residence that you can go and speak to get help. And then you can just get started.
There are more and more incubator spaces that just pop up everywhere. So that's also a way to try to get, for example, lab access if you don't have a university lab already.
Yeah.
You've worked on so many GPCR related projects, GPCRs. What were your aha moments as a scientist that kind of shaped your trajectory or refocus your interests on one GPCR versus another?
I think, the first aha was actually, funnily enough, it was actually while I was away on maternity leave, having a baby. I was in the neuroscience group, but while I was away, there was a reorganization in the department. And when I came back, that's when I had the opportunity to join the receptor systems team and focus on the cloning and identification of GPCRs. And, that led to a lot of the work, and particularly, the GABA-B heterodimer identification was a perfect aha, because we knew that we'd actually cloned a receptor, but we didn't really know what it was doing. The first GABA-B receptor, which is now called GABA-B1, we tried to express it and get it to signal. It just didn't work at all. So we were looking for some interacting accessory protein that we thought would help it. And we ran a screen to look for that. And then we pulled out the other GABA-B partner, the GABA-B2 receptor. And so when we were literally looking down the list of the partners that we pulled out on that screen, a lot of them were intracellular proteins, like you might expect, scaffolding proteins, this sort of thing. But then just literally looking down the list, there on the list was the second GABA-B receptor. And so that was like a real aha. But then it was made better because we immediately thought, this could be a heterodimer. And that was before any other heterodimers had been identified. So we did the classic experiment in Xenopus oocytes, where you inject the cDNA of both the receptors together. And we did that. And I was in the room when we then added on baclofen, the agonist, and we could see the ion channel activation. So that was a great science aha. And I did then spend quite a long time working on GABA B as a drug target, but that never actually worked out because we thought there were going to be lots of subtypes, which there are for the metabotropic glutamate receptors. We thought there would be multiple metabotropic GABAergic receptors, but in fact there weren't. And so it was very difficult to get a drug that didn't have side effects. So that was one aha moment. Obviously, I told you the other one, which was when I went to the LMB and met Chris Tate and Richard Henderson, and that sort of seeing their technology, I knew that would make a good company. And when all the time I was at, Sosei Heptares, we had many ahas every time we got a new structure. And it was particularly exciting when we got the first class B receptor structure, which was the CRF1. Nobody had ever seen a class B receptor before. And we had this really cool room where you could see, you could put on 3D glasses and see the receptor in 3 dimensions sort of floating in the room and moving around. So you could explore it in that context. So that was, a fantastic, fun time in my career.
A little bit out of a movie.
It was, yeah.
And the GABA B receptors were one of the first receptors I studied, and I haven't worked directly on them, but learning about them, I thought it was a really neat system that you actually needed the second partner to get on the surface and to signal.
Yes.
A really cool, really neat system. Were there any other moments that you thought were worth mentioning today?
Yeah, there are so many. I mean, some other ones was when we were trying to find the ligands for the free fatty acid receptors. And we were screening those in yeast cells because they don't have a background, so they're quite a clean system. And we thought they were peptide receptors, so we were trying to screen peptide libraries. And we got some peptides that activated them, so we got very excited. Then we tried to make lots of peptide analogues to get SAR, and it just made no sense. Some peptides were active, some were not active. It was very crazy. And then Andrew Brown, who was the person who was in the lab doing it, he just sat down with a table of what was active or inactive. And then he realized the peptides that had been dissolved in acetic acid or acetate were the ones that were active. And the ones that were dissolved in something else, the ones that were just used as basic ones, were inactive. So it turned out that the ligands were not the peptides at all. It was the acetic acetate. And then, of course, that became the acetate and butyrate activated receptors. So that was another fun aha moment.
That's really neat when you have to go back and look at what is your receptor we suspended in and realize that actually it's not the peptide.
Exactly, just the basic conditions of the assay.
Phenomenal. Before I let you go, I want to ask you one last question. This is a topic that has come up lately. Unfortunately, we have to talk about this in 2020, but what are your thoughts on increasing diversity in the field, not only in an academic setting, but also in a biotech and biopharma setting?
Well, first of all, it's so important that we do increase diversity in all its forms, gender, race, culture, etc. Because, I really know from my own experience that if you can put together a diverse team, you get a much more productive and happy team. If you get people, the problems we're trying to solve in science are really difficult and complicated, and they're not obvious. And everybody comes with a different perspective and a different way of thinking about it. So the more that you can get people with diverse attitudes and backgrounds and diverse ways of thinking about things, the more likely you are to come up with good ideas or ways of solving problems. So how can we actually do this? Well, I think one way is to try and have more role models that can set good examples. And I think, we've done pretty well in the sort of getting a better gender balance in the field. And that's because there have been, good women who've been, leading labs and setting examples. So I think we should be able to do that, in other aspects of diversity. But, it does fall down to people who are organizing conferences to really make sure they have the best possible diverse speakers, not just the same people who are there over and over again. I mean, the slightly disappointing thing is, having worked in GPCRs for 30 years, and I look at the conferences, the same people are speaking now as were speaking 25 years ago. So can we now try and encourage the next cohort of people to be the main speakers at conferences? I think it's so important. And, you know, people should actually, the old guard should decline to be speakers and actually put forward younger people from their labs to represent them and speak about their lab work. You know, it's important for lab heads and anybody who's got the responsibility to recruit people to make sure that they try and find diverse groups for their own labs, and then mentor those people as best that they can and promote them. So I think that has to come partly from the top down, and you know, people from diverse backgrounds hopefully should start to feel more empowered, that they can go out and search for opportunities and contact people and just say, here I am, this is what I have to offer.
You've touched on a very important point talking about mentorship. I think mentorship is very important at all levels, and making sure that people who work with you or for you feel supported, but at the same time, as a mentee, being able to ask for help and to reach out is very important as well. And you're right about the conferences. You look at the programs, it's always the same people. And that's why we're organizing with Dr. GPCR a new GPCR virtual summit, where anyone can submit an abstract and anyone can come in and make a presentation. And I'm encouraging a lot of PhDs and postdocs to come in and present their work. And even on the podcast, we're looking at having more postdocs come on in and present and talk to us about their work that they're doing, because they're the ones who are at the bench doing the heavy lifting.
No, that's great. I think that's a fantastic idea. It's
fantastic. Thank you so much, Fiona, for your time. I really appreciate it.
I really enjoyed talking to you, and I love the Dr. GPCR website and the podcast. It's a great idea, and I hope it will stimulate more and more people to work in this field.
I really hope so. My goal is to have as many people as possible reach out to us, have us contact us, and so that we can all be under the same umbrella. And if anyone needs any help, whether it's from biotech or academia, we can work together. You mentioned previously that at Heptares you maintain this close collaboration with academics, and you kind of evolved during that time, and you're still doing it, and that is amazing, and that's important. All right. Thank you so much.
Bye.
Thanks. Bye.
I need a favor. Please share the Dr. GPCR University program with your friends and colleagues, especially if they live and work in a developing country. Please subscribe to our YouTube channel. Many of you come back to watch our videos, but aren't subscribed. Having more subscribers will help us get you more content. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest and our Dr. GPCR team, Attila, Ines, Montserrat, Ivana, Cam, John, Yatsu, Andreina, and Balint. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. And don't forget to subscribe to our monthly newsletter on our website to get all the news about all the work that we've been doing and we are doing at Dr. GPCR directly delivered into your inbox. Another great way to support us is to share your favorite Dr. GPCR programs with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com or hello@drgpcr.org. As I mentioned, both emails will be active for a foreseeable future to make sure that we don't miss your email. Until next time, stay safe.